Hello everyone, I'm here with another video consisting of DBT-GRF previous year questions on proteins. So let's start solving the questions. The first question, it says the amino acid that can act both as an acid and a base in an enzyme catalyzed reaction is. So before proceeding towards our option, let's see this structure. This structure is of histidine molecule. Histidine, it's a basic amino acid due to the presence of imidazole ring, and whenever this imidazole ring is protonated, that is, it consists of the hydrogen atom, it acts as an acid. But whenever the imidazole ring is deprotonated, it acts like a base. So our correct answer would be option C, that is histidine. Next question is, which of the following amino acid changes in the active site of an enzyme is most likely to change its activity? Our options are lysine to arginine. There would be no change in the activity because both are positively charged amino acid. Then next is the leucine to isoleucine, no change since both are non-polar amino acid. Then next is glutamic acid to aspartic acid, no change since these are negatively charged amino acid. And the next one is lysine to proline. Lysine is a positively charged amino acid and proline, it's a non-polar. So there is a change in the positively charged amino acid to a non-polar amino acid. Therefore it would change the activity of the enzyme. Therefore option D is the correct answer. And next question is, phosphorylation of proteins is a typical post-translational modification which modulates their activities. Which one of the following amino acid residues can be phosphorylated? Always remember there are three amino acids, that is serine, then threonine and tyrosine, which are phosphorylated. But beside this, aspartic acid, glutamic acid, and in some cases arginine and lysine can also get phosphorylated, but among these aspartic acid mostly get phosphorylated. Therefore from the given option D is the correct answer. Our next question is, a bacterial culture grown in a medium containing radioactive sulfur would incorporate the radio label in the tetrapeptide. So there are two amino acids, that is cysteine and methionine, which contain sulfur in them. Therefore from the tetrapeptide you can see that option A contains the cysteine and methionine. Therefore option A is the correct answer. The next question is, a peptide of sequence serine, histidine, glutamic acid, leucine and arginine is isolated from bacteria. Which one of the following options lists the possible phosphorylation site in this peptide? Okay, in case of bacteria, besides serine, threonine and tyrosine, there is an additional amino acid, that is histidine. It gets phosphorylated. So from the given option A. The next question is, with reference to protein precipitation by organic solvents, which one of the following statements is correct? The organic solvents like acetone, methanol, what they do is that they decrease the dielectric constant of the protein molecules. As a result, the different molecules, they come close to each other and it decreases the solubility of the protein in a solution. Therefore our option B, that is it is dependent on the change in dielectric constant, is the correct one. Next question is, a young scientist was interested in creating a dipeptide using L-alanine and L-glycine. How many different dipeptides can be generated? So there could be a different combination, usually a protein or dipeptide. It consists of an amino terminus end and a carboxyl terminus end. So therefore by using alanine and glycine, there could be different combination. Either alanine can be on the amino terminus end and glycine on the C terminus end, that is the carboxyl, And next combination, the glycine can be on the amino terminus end and alanine on the carboxyl terminus end. Or it can happen that both alanine can form the dipeptide, and or in the same both glycine can form the dipeptide. So a total of 4 combinations are possible. The next question is, which kind of post-translationally modified protein targets are recognized by bromodomain containing proteins? Here bromodomains, these are the protein domains which recognizes the acetylated lysines. These acetylated lysines, they are present on the histone molecules. So our option A, that is acetylated protein, is the correct answer. Next question is, which one of the following is an achiral amino acid? An amino acid has a general structure like this. It has a carboxyl end, it has an amino end, it has a R group and a hydrogen atom. So whenever the 4 constituents around the center carbon atom are different, then that molecule is said to be chiral. For example, this is the structure of a alanine molecule. Here you can see that the R group is a methyl group, therefore it is a chiral atom, chiral amino acid. But in this structure you can see it's of glycine. The R group is a hydrogen atom, that is the 4 constituents are not different, therefore it is a achiral molecule. So from the given option B, that is glycine, is the correct answer. And always remember, except glycine, all amino acids, they are chiral in nature. Only glycine is a chiral amino acid. The next question is, which one of the following amino acids has a strong tendency to disrupt alpha helix and beta strand? This is the structure of a proline. The proline, it doesn't have a free amino group, that is this one, it doesn't lack. This amino group, it is involved in the formation of this cyclic structure, and this cyclic structure is the R group, that is the side chain, and due to the presence of this structure, it prevents the folding in a protein, therefore it disrupts the alpha helix and beta strand. Therefore the correct answer is option C, that is proline. The next question is, which one of the following is not a characteristic of a collagen fiber? Let's read the options. The first option is, each of the strain is independently stable due to the presence of hydrogen bond. This one is the only wrong one, because the collagen fiber, it consists of a triplex molecule, means triplex helix, in which these 3 are stabilized by different intermolecule and intramolecular bonds. Therefore our option A is the incorrect one, that is, according to the question, it is not the correct answer for the collagen fiber. The next question is, a protein X is active when phosphorylated on threonine residues. You wish to mimic this phosphorylation by mutating threonine to another residue. Which one of these residue will you mutate threonine into? As I have already said that there are 3 amino acids, that is serine, threonine and tyrosine, that are phosphorylated. Therefore if you mutate threonine into any of them like serine or tyrosine, it will continue to phosphorylate. So from the given option you can see that is option D has the tyrosine, that means option D is the correct answer. The next question is, which one of the following is used to validate the secondary structure of protein? It's a straightforward question. Therefore option C, that is Ramachandran plot, is the correct answer. So in this way our question round ends. Hope this would be helpful to you, and all the very best for your exams. If you want more questions to be solved, please do let me know in the comment section. Thank you.